This is online lecture 18 for Bio 240, and this week we're going to talk about population genetics. So population genetics is the study of allelic frequencies within an entire population instead of just within individuals. It's also the study of the change over time within a population. In other words, evolution, how evolution occurs within populations. So to start with, let's talk about what is a population in the study of population genetics. And geneticists who are studying populations refer to their groups as Mendelian populations. And this basically means that it's a group of interbreeding individuals. So it doesn't apply to yeast or fungi or some organisms that reproduce asexually. And they have a common set of genes. And this is referred to as the gene pool. And again, what population genetics is really the study of allelic frequencies within a population. So why do we need to study this? What's useful about it? Well, there's many situations in humans where this is particularly important. For example, you might want to know the frequency of disease alleles. For example, what's the frequency of a certain allele for the cystic fibrosis gene within a population? And we know that those disease alleles are more frequent in certain ethnic groups than in others. So being able to calculate this can be really important. Another example is maybe we want to know the frequency of resistance alleles. And again, remember that in sickle cell anemia, there are some populations that have only one sickle cell trait, and they are resistant to malaria. So maybe we would want to calculate the frequency of those alleles within a population. Or there are certain alleles to the HIV receptor on white blood cells which confer resistance in select groups of people, and figuring out the ratio of those within a population could be really important. Another reason to study population genetics would be to look at the frequency of alleles in ecology. For example, by looking at the frequency of alleles in a population, you can tell whether a population is changing due to migration of individuals in or out of it. So in studies of rainforest destruction, looking at ecological changes is really important. And then finally, by no means inclusive list, there have been population geneticists who study bioterrorism. So these scientists are able to use population genetics to identify where certain isolate came from. For example, when the anthrax attacks happened, where did those anthrax strains originate from? And population genetics was able to inform that study. So today we're going to focus on three things in our discussion of population genetics. The first is how to calculate the frequency of genotypes within a population. Then we'll talk about how to calculate the frequency of alleles within a population. And finally, we'll talk about how to calculate the expected frequency of alleles and gene in the next generation. And some of this will probably be familiar with you from Bio 181, 182 when you learned about the Hardy-Weinberg equation. So that's what we'll talk about today. On the next lecture in population genetics, we're going to focus more on the implications of the Hardy-Weinberg conditions and how that can lead to evolution. Let's begin by talking about a simple situation with the MN blood group. In humans. And there are just two alleles for this gene, the M allele and the N allele. And there's, from that, only 3 genotypes. You could have MM, MN, or NN. And remember, the M and N alleles are codominant. So that makes this really easy. When you do a blood test in people, it's easy to see what their genotype is by the expression of M, MN, or N antigens on their blood. So let's say we took a population of 100 people and we did a blood test to determine their MN blood group. And we found that 14 people had MM, 80 had MN, and 6 had NN. So from this data, it should be easy to figure out the genotypic frequencies. So pause the video right here and see if you can calculate the genotypic frequency for each of these genotypes. So you should have been able to get, for the type MM genotype, it's 0.14. That's simply 14 out of 100. So 14 divided by 100 is 0.14. We got 0.8 for MN and 0.06 for NN. Now we can use this information to calculate the allelic frequencies. So let's call the frequency of the M allele P and the frequency of the N allele Q. Since we only have 2 alleles in this population, P plus Q have to add up to 1. To calculate the frequency of the M allele, we're going to need the total number of M alleles in the population divided by the total number of alleles in a population. And then Q would be the same thing, but we only really have to figure out the frequency for one, and then using this equation here, we can subtract to find the other frequency. So let's focus on M first. So going back to our population here, we have 14 people that have 2 M alleles. So we have 14 times 2. Then we have 80 people who have 1 M allele. So we have plus 80 times 1. So those are the total number of M alleles in the population. And then we have a total of 100 people, each of which have 2 alleles. So the total number of alleles is 200. So we calculate that P equals 0.54 for this population. Now pause the video here and see if you can do the same calculation this way for the Q allele. So I've drawn out the same calculation for Q using that population. The other way we could have done it is to say P plus Q equals 1. So we have 0.54 plus Q equals 1. Solving for Q, 1 minus 0.54 would give us 0.46. Now that we've calculated the allelic frequencies in our population, what if we want to predict the frequency of each blood type in individuals in the next generation? So our allelic frequencies actually also tell us the gamete frequencies. Since one allele gets distributed to every gamete, so we would have a frequency of M. Remember, M was P equals 0.54, and for N, Q equals 0.46. So those are the frequency of the gametes in this population who are going to have the M or the N allele. And now we can predict the blood group by drawing out a Punnett square. So if we have M, N, and M, N, and when you're drawing this Punnett square, try not to confuse it with what we talked about at the beginning of the course. So we're not talking about an individual who's MN crossed with an individual who's MN. We're talking about a population in which there's 0.54 frequency of the M allele and a 0.46 frequency of the N allele, since there's just two N alleles, breeding within the same population. So we have the same frequencies. So to calculate what this would be, to have type MM would be 0.54 times 0.54. MN would be 0.54 times 0.46, 0.54 times 0.46, and then finally for the NN, 0.46 times 0.46. And notice that this is P squared. This is PQ. This one is Q squared. And this one is PQ. So the expected frequencies in the next generation for MM, expected frequency is P squared, which equals 0.54 times 0.54, 0.29. For MN, the expected frequency is 2 times PQ. So PQ is 0.54 times 0.46 times 2 gives you 0.5. And then for NN, the expected frequency is Q squared, 0.46 times 0.46, which equals 0.21. And remember, since these are the only genotypes possible in this population, a good check is to go back and make sure that these all add up to 1. So we have 0.29 plus 0.21 plus 0.5. Those all add up to 1. And this brings us to the Hardy-Weinberg equation. P squared plus 2pq plus q squared equals 1. So that's the prediction for the frequency of alleles within a population. So the Hardy-Weinberg law states that under a certain set of conditions, which I'll describe in just a second, the genotype frequencies are P2, 2pq, and q squared when there are 2 alleles for a gene within a population, and that these all have to add up to 1. So that allows you to calculate the frequencies of alleles within individuals in the next generation. But again, there's several conditions under which Hardy-Weinberg applies. So the assumptions of the Hardy-Weinberg law are that there are no new mutations that might increase the frequency of an allele. Another one is that there's no migration of individuals in or out of the population. So it's a closed population. We assume that it's a large population. So it's easy to do an example with 100 individuals, but 100 is actually not a large population, so we wouldn't meet the preconditions for the Hardy-Weinberg equilibrium. The fourth condition is that there's random mating. Between individuals. So for example, with the blood type, most of you don't see blood type, and so that wouldn't affect mating. People mate with each other not based on their blood type for the most part. So we could assume that there would be random mating between MM, MN, and NN individuals. And then finally, no preferential survival for any one blood type or genotype. So it is the Hardy-Weinberg law assumes that for any given genotype, the survival advantage would be the same. So when these conditions are met, the population is said to be under Hardy-Weinberg conditions or in Hardy-Weinberg equilibrium. And that allows us to use this equation to be able to calculate the expected allele and genotypic frequencies in the next generation. And we'll have a chance to practice this in class on Thursday.